Hello students, this is Dr. Faizan Mirza. In this video, I'll be discussing with you the use of chi-square test, and the question that I have taken here is from May-June 2021, variant 51. The question here begins with you are given some information, which means that the content already will be provided to you, all the information related to what genes are controlling what traits and how many alleles are there for those genes. So everything will be provided to you. For example, in the question they say inheritance of flower color and flower position. These are two traits. One is flower color and the other is flower position. In the pea plants controlled by two genes. So these two genes could be on the same chromosome and they may be linked, and they may be on separate chromosomes and they may not be linked. So we do not know as far as these two genes are concerned. So gene P and small p control flower color. Capital P is for purple and it's dominant, where small p is for white. So we know that all purple flower must be having the dominant capital P, and the white ones will always be small p, small p. Same goes for gene AA, controls for the flower position. Capital A is for the side of the shoot, which is axial, and small a is end of the shoot. So all capital A, capital A, and capital A, small a will have axial, whereas small a will be terminal. So always the information provided to you, you just should annotate what those alleles mean, so that it becomes easier. So this here you have they actually have told you what exactly is the terminal position and what you mean by axial position of the flower. Now, we know that when we do the crosses, what they have done about this predicted that if the genes are on the different chromosomes, the ratio of phenotypes of F2 generation would be 9331. What do? We cross purebred. We cross a purebred dominant, and we cross a purebred recessive, and we cross them together, we get the F1 offspring. The F1 offspring will always be hybrid, and once we have the hybrid offspring, they will be 100% hybrid. Now, these 100% hybrid offsprings can be crossed together. So F1 into F1 offsprings, two F1 offspring can be crossed together, and we'll get the F2 phenotypes. If the genes are following independent assortment, then F2 generation will have the ratio of 9331. There will be four phenotypes. So this information is already provided to you. There should be four phenotypes. So the table, the results for F2 generation is shown here, and you can see the F2 phenotypes. These are mentioned to you here, and the frequency is provided to you. Frequency means the number. When you add them all together, this comes out to be 3345. Now, what do? This frequency provided to you is observed, and expected. How do you calculate the expected? We divide this in 9331 ratio. So 9 over 16 into the total, 3 over 16 to the total, 3 over 16 to total, and 1 over 16 to total. So that will give us the expected value. So what do? The next they are asking you, why is the chi-square test used? So it's used when the data is discontinuous. We know that. State the null hypothesis. Null hypothesis is there is no significant difference between the observed and expected ratios when F1 offsprings are crossed together, because they are asking for the null hypothesis. Next, they are asking you to carry out the chi-square test. So already we have calculated the expected values. The expected values means you need to just multiply the total with the fraction of that particular phenotype that you expect according to 9331 ratio. In this, it's preferable the expected values should be in whole numbers. You should just round this off accordingly, so you get the answers in the whole number. Then we know that our first column is O. Then we need to get O minus E, which means we will subtract the value 182 from 1756, we'll get O minus E. Then we'll take the square of that, and O minus E squared over E will be our answer. And we'll add this answer in the last column, and the answers will be placed into DP here in the last column. When we add up the last column together, we get the summation of chi-squared values. Here in this case, 21.48. Now, the next is we take out the degree of freedom. So degree of freedom, we know in chi-square test, we have to take out the degree of freedom by n minus 1. 4 minus 1 is 3. So this free degree of freedom here in probability level will be taken 0.05. The critical value is 7.82. Now, if the critical value is 7.82 and our chi-squared is greater than 7.82, so we know there is a significant difference, and the null hypothesis will be rejected. The null hypothesis will be rejected in this. What can be the conclusion? The conclusion will be that both the genes capital A small a and capital P small p, they do not fall into bond assortment, and the genes are following most probably linkage. So that's all from my side. That's it. I hope the video is helpful for you. Thank you so much.